Good morning, everyone. I am Dr. Jyoti Batra, Dean Research and Professor of Biochemistry. Today, we are going to talk about a very important biomolecule of the body, that is carbohydrate chemistry. Uh, as per competencies, as per given by the NMC, for the chemistries, we have to discuss and differentiate monosaccharides, disaccharides, and polysaccharides, giving examples of main carbohydrates as energy fuel, structural element, and storage in the human body. And the second competency says about describe the processes involved in digestion and assimilation of carbohydrates and its storage. Now, why carbohydrates are important? This is chief source of energy because it can go to the brain anytime and gives you the ATPs if required. Important structural components of cell membrane, important part of nucleic acids and free nucleotides and coenzymes. Major antigen and are carbohydrates in nature, for example, blood group substances. Biological role as a part of hormones and their receptors and enzymes. If you remember, when we talk about nucleic acids and free nucleotides, RNA and DNA, the name suggests that there is a pentose sugar, that is ribose, and that is present in RNA, and deoxyribose, that is present in DNA. When we go for the vitamins B complex, which acts as coenzyme, the structure of these vitamin B has a lot of carbohydrate components to its structure. This is the empirical formula of carbohydrates, that is CH2ON, and that's why these are called hydrates of carbon. And N suggests the number. If carbon, it's a 3 carbon compound, N will be 3. If it is a 6 carbon compound, it will be 6, and so on. Now, what are carbohydrates and why it is important for a medical student to study it? Carbohydrates are having free aldehyde and keto group, and they are very strong reducing agents. A medical student should know about their importance because you have heard a lot about diabetes, you have heard about fasting and starvation. So, how it is associated with this molecule? In this lecture, we will first know how this molecule is metabolized, what is its structure, and how it later we will come to the point where its role comes as a diabetic agent. Now, as I said, carbohydrates have free aldehyde and keto group. Aldehyde group, as you can see, we have taken C3 compound, glyceraldehyde and dihydroxyacetone. Aldehyde group is shown in the glyceraldehyde and ketonic group is shown in the dihydroxyacetone. Their molecular formula, their formula is same, chemical formula is same, but their structural formula are different. So in a way, these are an isomers to each other. Now, what are different types of monosaccharides? As I earlier told you, depending upon the number of carbons, we can call them trioses, tetroses, pentoses, hexoses, heptoses, and nanoses. So 4 carbon is erythrose, 5 carbon is ribose, 6 carbon is hexose, that is glucose, which we are going to take up, and very important molecule. Then heptoses, pseudoheptulose. 9 carbon is neuraminic acid, which you are going to study a lot in heteropolysaccharides. Now, disaccharides after monosaccharides, when two monosaccharides can be linked by a bond, that bond is known as glycosidic bond, which is very important. You can see that how this glycosidic bond is synthesized. We can see here one molecule of galactose and another molecule of glucose. It is attached by carbon 1 of galactose to carbon 4 of glucose. So the bond which is being there is galactosyl beta 1,4 glucose. This is called the glycosidic bond. Any two carbohydrate monosaccharides moieties joined together to form a disaccharide, oligosaccharide or polysaccharide has to have glycosidic bonds. We will discuss about different type of glycosidic bonds at the end of this chapter. Now oligosaccharides are 3 to 10 units, polysaccharides when the units are more than 10. Examples of disaccharides. Let's talk about disaccharides. As we have already discussed that sugars can be reducing or non-reducing. But as a rule, all the monosaccharides has to be reducing sugar because they are single moiety and their reducing group or their functional group is free. There is no attachment to that. But in the case of disaccharides, there are 3 examples I am going to show, out of which 2 are reducing and 1 is non-reducing, and we will see why these 2 are reducing and why this one is non-reducing. First, let's talk about the reducing one. As we have already discussed that reducing property of the glucose or the sugar is because of the presence of aldehyde or ketonyl group, which is present in the aldehyde sugars at carbon 1 and in the ketonic sugar at carbon 2. So in a disaccharide, if carbon 1 and carbon 2 or both or either of them is free, the sugar will be known as reducing sugar. But if carbon 1 and carbon 2 are associated in a bond formation, the sugar will become non-reducing. When we see the lactose, which is a combination of galactose and glucose, as you can see in the diagram that what is happening here? C1 of the galactose is attached to the C4 of the glucose. So once this disaccharide lactose is formed, it has a reducing property because of the carbon 1 which is free in the glucose molecule. Because what we have seen that carbon 4 has been associated for the bond formation. So C is free, C1 is free to give lactose its reducing property. Similarly in the maltose, same, there are 2 glucose molecules which are associated by beta 1,4 bond, and one glucose has shared its functional group, the other's functional group is free. So that is why when maltose is synthesized by 2 glucose molecules, it still becomes a reducing sugar because we have one functional group which is free at second glucose molecule. However, in the case of sucrose, it is little different. What is happening here? Glucose and fructose are associated by carbon 1 and carbon 2. Carbon 1 is aldehyde sugar glucose and carbon 2 is ketonic sugar fructose, and they are associated at both the functional groups. The bond, glycosidic bond, is between 1 and 2, C1 of glucose and C2 of fructose. That is why both the functional group is gone for the bond formation. It is a non-reducing sugar. Now, sorry, this slide has to come here. Then comes isomers and epimers. We can go back to this slide also. Isomers and epimers are actually formed when we talk about the stereoisomerism in the carbohydrates. That has been shown because there are different type of isomerism which is seen here. First we'll talk about isomers. Isomers are the compounds with the same chemical formula but different structure. For example, glucose, mannose, fructose, galactose. As you can see in the picture that glucose and mannose are the isomers to each other. Similarly, even glucose and fructose are isomers to each other. Epimers, isomers that differ in configuration only around one specific carbon atom. Then they become epimers. If we take the example of galactose and glucose, you can see in the blue that OH at the carbon number, around the carbon number 4 has different orientation in both the groups. That's why they become C4 epimers of each other. galactose and glucose. Similarly, glucose and mannose, you can see in the red that the orientation around the carbon number 2 of OH is totally opposite. So they become the C2 epimers to each others. Now enantiomers. Another type of isomerism is known as enantiomers, which are always the mirror image of each other. Two members are designated as L and D isomers. As you can see in the picture, it seems that there is a mirror there. And the right hand side image is known as D-glucose and the left hand side image is known as L-glucose. And humans have the enzyme which recognize a specific enantiomer. Basically, we have more of D form rather than L form. However, this DLL can be converted by specific isomerases. For example, glyceraldehyde can be converted to dihydroxyacetone in the presence of an isomerase. So these become. Now, what is a racemic mixture? Why we have to study about monosaccharide cyclization and how anomeric carbon is important here. When we dissolve a powder of carbohydrate in the water, it becomes a solution, and as soon as it comes into the solution, it rotates the light. The rotation of the light changes with the time. Okay. Why? Because carbohydrate exists in anomeric carbon of the carbohydrate, that particular carbohydrate exists in two forms in solution. One is ring or cyclic structure. Another is what happens in the ring and cyclic structure. CHO of the aldehyde sugars or the ketonic group of the ketonic sugar reacts with OH group of the same sugar, making the carbonyl carbon asymmetric. Okay. So 99% in solution we have this kind of structure. Asymmetric carbon is the anomeric carbon. Second is acyclic or open chain, which is less than 1%. So as you can see in the two different diagrams, one is showing you the ring structure, another is showing you the open chain structure or acyclic structure. In the ring structure, you can see that there is an anomeric carbon. Anomeric carbon, the carbon bond at the anomeric carbon opens and is converted to, you can see the orientation is changed. H comes to the place of OH and OH goes to the place of H. This happens when it is into the solution, and this is called alpha form and beta form. Alpha form is when it is towards the ground, beta form when it is above the ground. Okay, so these combination of these two in a mixture is called your racemic mixture. Similarly, in the open chain structure, you can see that there is a bond formation between the aldehyde group and the OH group. And what is happening? There is a cyclization of the beta and the D form. In the blue, you can see the orientation of OH is on the left side, and in the red form, you can see that orientation of OH is on the right side. So right side orientation is always alpha form, left side orientation is beta D. So because of this generation of asymmetric carbon, there is a new pair of anomeric carbon and isomers are generated, and these are known as alpha beta configurations. Alpha D glucopyranose and beta D glucopyranose, which you have already seen. Now these are different from enantiomers because these are not mirror images. For example, glycogen synthase recognizes alpha D glucopyranose while cellulose is synthesized from beta D glucopyranose. So now you understand the difference. Glycogen can be broken down and stored in the human body, but cellulose cannot be, because our body doesn't recognize the beta. Our body doesn't have the enzyme to break down the beta bond. It has the bond. It has the enzyme to break the alpha D bond. Alpha and beta forms of sugar solution slowly and spontaneously form an equilibrium mixture, which is through the mutarotation process, and this is known as racemic mixture. This is known as racemic mixture. Now reducing sugars. Sugars where anomeric carbon is free and not linked to another sugar molecule can act as reducing sugar, which we have already discussed. So all monosaccharides are reducing sugar in disaccharides except for sucrose. Others are reducing sugars. How do we identify the reducing sugars in a sample? Suppose if you want to test the urine sample for a reducing sugars presence, we will put the urine sample into a reagent, which is a chromogenic agent. In this case, we put the Benedict reagent and we heat it, and then with the heat there is a reduction of chromogenic agent, and it gives a color reaction or color solution or the precipitate. Depending upon the color of the precipitate, we can semi-quantitatively tell the percentage of sugar in the urine. So what is the clinical interpretation? This is the colorimetric test of the abnormal urine sample for diabetes mellitus. Okay, if it is positive, that means a person is frank diabetic and the sugars concentration in the urine is more than the concentration of sugar in the blood is more than 180 milligrams per deciliter, and now it is being excreted into the urine and the Benedict's test comes positive. So that is the clinical outlook, why we have to study these reducing sugars and how do we identify in the urine sample of a patient. Now coming to the glycosidic bonds, there are two types of bond, alpha and beta bonds. We have already discussed that. Bond that link sugars are known as we have two types of glycosidic bonds according to the number of connected carbons and position of anomeric hydroxyl group of the first sugar involved in the bond. If anomeric carbon is in alpha configuration, linkage is alpha bond. What do you need to break these bonds? Enzyme is glycosyl transferases. Substrate, nucleotide sugar, for example, uridine diphosphate glucose. When we will discuss about the glycogen metabolism, we'll talk more about it. And naming, as you have already known, alpha bond, when the anomeric carbon is in the alpha configuration. If anomeric carbon is in the beta configuration, it is known as beta bond. We have already discussed the structure of sucrose, maltose and lactose and the configuration as far as anomeric carbon or the functional group is concerned. Then types of glycosidic linkages can be OO types and NO type. When O and OH group are associated, we call it OO type, and when this is associated with non-carbohydrate component, for example NH2 group of any amino acid, these will be NO type. Now coming to the dietary carbohydrate digestion can be done by at the four levels. First and the foremost, everyone knows salivary alpha amylase, and that is why if we eat very fast or very hot and cold food, carbohydrates are not digested in the mouth properly because salivary alpha amylase becomes non-functional at high or very low temperatures. Once the food is going through after mastication salivary alpha amylase had functioned. It has broken the complex carbohydrate into polysaccharides into the disaccharides or smaller sugars. After that, pancreatic alpha amylase comes into the function, and that digests rest of the carbohydrates into the simple disaccharides and monosaccharides. Once it is there, there are some channel systems are there which help in the absorption of intestinal disaccharides and monosaccharides. Intestinal disaccharides are actually major of them are broken down into the monosaccharides, and these are absorbed in the body. What happens when there is abnormal degradation of disaccharides? And why it happens? It happens because of the digestive enzyme deficiencies. If the enzyme is deficient or it is not working properly, there will be a clinical condition, because that sugar, then the sugar will not be broken down, degraded into the monosaccharides for the digestion, and it will be creating clinical symptoms. For example, lactose intolerance. When lactase enzyme is missing from a person's system, what happens? Milk is not digested properly. A person has flatulence and abdominal cramps and pains, because lactose sugar is not being broken down due to the absence of lactase into glucose and galactose, and it is not being absorbed. Okay, so this is a clinical condition develops in case, and the patient has to be refrained from milk and milk products. Then comes the congenital sucrose isomaltase deficiency. Finally, its diagnosis. So this is how carbohydrate chemistry and absorption and digestion of carbohydrates is managed in the body. So what are my take-home points? That carbohydrates are aldehyde or ketones, anomeric carbons. They have lots of stereoisomerism, anomers, isomers, and enantiomers. What is racemic mixture? What are alpha and beta forms? What is capital L and D forms? What is pyranose and open acyclic structure? And finally, how it is digested and absorbed by different amylases. First is salivary and another pancreatic amylase, and then its absorption into the intestine. If there is some problem with the degradation of disaccharides, there can be lactose intolerance, which is a major kind of intolerance. Congenital sucrose and isomaltase deficiency can also be there. Next, in the next class, we will be talking about the metabolism of carbohydrates, how glucose is metabolized, how fructose is metabolized in the body, and from that metabolism, how do you get the ATPs? That's the final goal. And if there is some problems in these cycles, in systems, in metabolism of and biochemistry of these molecules, what clinical conditions can develop and how to manage those clinical conditions, what are the symptoms and what are the biochemical parameters that has to be measured when these kind of clinical conditions are there. Thank you so much.